the session here, and let's start talking about the nervous system. When we start talking about the nervous system first, before we get in, before muscles, I know that in the book, it's chapter 10 is muscles and chapter 12 is the nervous system. But I really feel like when we do the nervous system first, it helps because there is some overlap of information and concepts when we talk about muscle. So we found out that the nervous system is just easier to start off. So what is the nervous system? What is the general function of our nervous system? Okay, way back in chapter 1, we learned about the homeostatic mechanism, how you have a stimulus, then a receptor, then a control center, and an effector. And so our nervous system is our communication system for a lot of that homeostatic system. In fact, it is the control center portion. Right, so the control center is going to integrate that information. It receives information, input we call it, like a computer, and it will then integrate that information, analyze it, and then decide how it's going to respond. And that's basically what we're going to see, right? So when we're looking at the nervous system, yes, it's a communication and control system, right? But it's going to pretty much oversee all the functions that are occurring in your body. Right, down to the most minute, like basal metabolic rate. That's kind of like the upkeep, the basic functions that cells and tissues perform, like when you're sleeping, right? And it does a lot of these functions through the use of electrical activity. And so as we progress through this chapter, we're going to learn about all these different functions and how it does what it does. So let's start off by talking about functions. What's its first function? Collect information. What's going on outside our bodies and inside our bodies. We're going to have, right, stimuli, which is going to be a change, all right, in either the internal or external environment. So we're going to have these stimuli occurring. And so we needed a way to detect that stimuli. So you have these special receptors all throughout your body, both, that are going to monitor any changes inside and outside. And so they're going to send that information, when they're activated, these receptors, and they're going to send that information to your central nervous system, your brain and your spinal cord. And it does so through what we call sensory input or sensory signals. So it's going to send that information to your central nervous system. And then your central nervous system, your brain and spinal cord, are going to figure out how they're going to interpret that information and respond to that information. And so it'll process it, evaluate it, and then finally decide, okay, now we figured out what's going on. This is how I want to respond. So it's going to initiate some sort of response. And so your control center, your central nervous system, is going to send information to the effector organs, which are muscle and glands. And it'll do so through what we call motor output. Okay, so sensory information is input and motor information is output. Okay, so a lot of these terms, if you've taken a computer class, it's very similar, right, to how computers work and CPUs work, right? This is a very similar concept here. All right, so when we break down our nervous system, we're going to break it down both anatomically, which is going to be structurally, and then we're also going to break it down physiologically, which is the function. So let's start off by looking at the anatomy or the structural organization of our nervous system. And so we break it down into two parts: the central nervous system, which you've heard me mention many times, which is the brain and the spinal cord. And then we have the peripheral nervous system, which is everything else outside of the brain and spinal cord. So that includes what we call nerves and ganglia. And we're going to go through some of these definitions in more detail. Okay, but for right now, from an anatomical standpoint, okay, our nervous system is broken down into two parts: central nervous system, which is the brain and spinal cord. And then we have the peripheral nervous system, which are going to be nerves and ganglia. And we'll talk about what nerves and ganglia are. Okay, so keep in mind, so nerves, you've heard of some of the nerve names before. Oh, my sciatic, I got sciatica. Well, that's the longest nerve, the biggest nerve in your body. All right, and it's just a bundle of axons, and we'll go through all these different structures. And then just think of ganglia. That's going to be where there's a cluster or a grouping of cell bodies. We'll talk about the significance of what happens at the cell bodies. Okay, so the other way that we are going to organize our nervous system is the physiological way, or the functional organization. So when we're dealing with that, right, it's a little bit more to it. So we have what's known as our sensory nervous system, and then we have our motor nervous system. So like I said previously 2 slides ago, we kind of compare this to a computer. You've got your sensory input, and that's all that information, right, that the sensory nervous system brings in to our nervous system, all right, our central nervous system. And then we have the output, which is where the motor nervous system kind of takes over. So we break down our sensory nervous system into 2 parts. You have your somatic sensory and your visceral sensory. So the somatic sensory is all the sensory input and information that's coming in that you can consciously perceive. So your 5 senses, taste, all right, sight, hearing, all right, that is somatic. Okay, you can consciously perceive those, all right? Temperature, okay, it gets cold, all right, pressure, deep touch, light touch, right? Those again are part of the somatic sensory. So if you have conscious perception, we're dealing with the somatic sensory portion of our sensory nervous system. Now the visceral sensory, think of viscera, organs, right? So this sensory input is not consciously perceived. So you don't really notice when your heart rate goes from 66 beats per minute to 68 beats per minute. Or let's get crazy and say 70 beats per minute. You're not really conscientious, or you're not really aware of when your blood vessels vasodilate or vasoconstrict by 10 to 20%. You don't know that, right? So that falls under the visceral sensory, all right, nervous system there. All right, so that sensory information, all right, is going to be below our conscious perception. We're not really going to notice, all right, unless it gets really bad, if our partial pressure oxygenation rate in our blood goes from 96% saturated to 95. Okay. Now, if it drops significantly, then there's other ways to figure that out. Okay, but that's our visceral sensory. Now, the other part of the functional organization of the nervous system is the motor nervous system. All right, the output, the motor output. So we break that down, okay, into 2 parts. We have our somatic motor. And to remind you, the motor nervous system is going to take, all right, the commands, the responses from the sensory nervous system and transmit it out to the effector organs. And so the somatic motor is the motor output that we can consciously or voluntarily control. And when we're dealing with the motor aspect, the only tissue that we can consciously and voluntarily control is skeletal muscle tissue. So that's an easy one. So somatic motor nervous system only is going to affect skeletal muscle. All right, so that's easy. The autonomic motor, all right, that's different. Autonomic is going to, again, deal a lot with our organs and our viscera. And so that is the motor output that we don't consciously or voluntarily control. Okay, so it is not consciously perceived and it is involuntarily controlled. And so that's going to be the other effector organs, which include cardiac muscle tissue, smooth muscle tissue, and glands. So all of that falls under the autonomic motor. So what are the 2 primary functional divisions of the nervous system? Sensory nervous and the motor nervous. Okay, so those are our 2 functional divisions. How do they differ? Okay, the sensory nervous system detects stimuli and transmits information from the receptors, all right, that were triggered by a stimulus, all right, to the central nervous system. Motor nervous system initiates and transmits information from the central nervous system to the effectors. Effectors are either muscles or glands. You should know that. Make sure that you input that into your brain. Okay. So now that we have kind of a little bit of an idea, all right, of the organization, both anatomically and physiologically of our nervous system. Let's get on and talk about some of the characteristic of the neurons. The neurons are going to be the nerve cell that transmits information, electrical impulses. So we should learn a little bit about some of the characteristics. So the first one listed up here is excitability. And basically the excitability is that this neuron, this cell, all right, has a responsiveness to a stimuli. So at that point, okay, prior to a stimuli, the neuron is at rest. But when a stimuli occurs, now that stimuli is going to change what we call the cell's membrane potential. And the membrane potential is, we'll get into it, it's a in-depth concept, but it's basically the electrical charge of the plasma membrane to a neuron. And so it's the ability for that plasma membrane's electrical charge to change. So that causes the neuron to be excitable. So we said before how the nervous system uses electrical signals, right, for its communication. And that's where conductivity comes in, right? It's the ability to propagate, all right? That's going to mean that we're going to maintain, or we're going to generate an electrical signal, and then we're going to maintain it. And so when we talk about conductivity, that's the ability to propagate the electrical signal. So we'll see that when we talk about the voltage-gated channels that we're going to learn about that are all throughout, all right, the plasma membrane here of our neuron. And so we're going to learn about where these gates are, what a gate is, all right? If you recall, all right, voltage-gated channels are proteins. They're transport proteins. And what they'll do is they're going to allow the movement of certain substances in and out of cells. And so we're going to see how these voltage-gated channels are going to allow the movement of certain substances in and out of the neuron. All right, the next characteristic is secretion. Okay, so we know when you're secreting something, usually you'll make it, not always, but you'll make it and then you'll get rid of it. You'll emit it out. And so when we're talking about secretion here for a neuron, we're going to discuss the release of a neurotransmitter. So we're going to see how an electrical signal is going to trigger the release of a neurotransmitter, and how that neurotransmitter is going to leave one cell and then affect a neighboring cell, another cell. So we're going to talk about how this occurs, how that happens, how the chemicals from one cell can affect and come in contact with another cell, and then how it influences what that other cell does, because it could be a gland cell, it could be a muscle cell, right? So we call that the target cell. So we'll learn about that. Neurons are pretty awesome because they live for a long time. And in fact, most of the neurons that you are born with, you will die with. You'll have them forever, right? So they have a long period of life, all right? So they have extreme longevity. Problem is that neurons are ameiotic, which means they will not undergo meiotic division, right? So what will happen is after, you have developed inside of mommy, you're born, then pretty much, now this is not absolute, but neurons are amitotic, they will not divide. Now there's always exceptions to the rule, we get into the amygdala and all that, okay? But for the most part, okay, neurons do not divide. All right, so here's a nice picture. I love this picture. Right, it shows a neuron and then another neuron. So let's swing it up here. Okay, you can see here's a neuron cell body. Then you have these processes that come off, and those are the dendrites. The dendrites will help receive or transmit information. Okay, then you can see there's one big, huge process that comes off. And that big process is what we call the axon. And you'll notice how the cell body starts to taper down, all right, into the axon. In the bone, we saw the epiphysis there taper down into the diaphysis, in that region we call the metaphysis. Well, here in our neuron, we see where it starts to kind of taper down, and that's called the axon hillock. Axon hillock is very important. I'm going to say it now, okay? This is where we start our action potential. Now, you don't know what that means, and that's the start of the electrical signal. But it's asked on questions all the time. Where do we initiate the action potential? Here at the axon hillock. All right, so once we've generated our action potential, our nerve signal, it travels down, all right, the axon, much like an electrical signal is traveling through the phone wires near your house, or the electric wires there near your house. So it travels down to the end of the axon. Sometimes you have what we call collateral axons, and they're just a split in the wire going somewhere else. And so the signal will head down to the collateral axon. It'll head down the regular axon, right, until it gets down here to the end. Right, and at the end, we have our terminal extensions here. We call those teledendra, these little kind of processes that are coming out. And you'll see at the very end a knob there, and we call those synaptic knobs. Now, these synaptic knobs will be contacting either another cell or an end organ, okay, or effector, which is a muscle or a gland. So you have three options. It could be another neuron, it can be a muscle, or it can be a gland. And so the synaptic knob will sit right on top. Let me show you. We'll zoom in here. Here you can see, here's our synaptic knob. And then it sits right on top of our target cell here. And we'll talk about what happens down in here. Okay, so you'll have these chemicals that are being stored here in the synaptic knob. They get released, and then they'll trigger, all right, something to occur, all right, on the other neuron or the effector. So through this chapter, we're going to talk about this stuff. We're going to learn about this stuff. All right. And, well, excuse me, I should rephrase that. We're going to review this stuff because you should have already looked this over. And so some of this should look familiar to you. Some of it might not, but some of it should. So if you go, let me go back to this slide here. All right. So you can kind of see here where this synaptic knob comes in close contact, all right, with the effector or neuron here. And so there's a small gap in between. You can see it. There's a small gap in between the synaptic knob and the other neuron or effector. So that's called the synaptic cleft. So the reason I mention that is because that brings up synapses. Okay, synapses are going to be a place or an area where a neuron is going to connect to either another neuron or an effector organ, which is, you should know this by now, a muscle or a gland. I'm going to keep saying that. Hopefully it will permeate into your brains and you'll never forget it. So we have two types of synapses. We have a chemical synapse and an electrical synapse. Now we're going to spend a lot more time talking about the chemical ones because they're just more common. Electrical synapses, we're not going to really see. We're only going to really see those, right, in some of the areas of your brain and pretty much your eyes. So there's really not much that, when I say exciting, was just good in this situation because it's less to have to memorize, right? But when we're looking at an electrical synapse, right, we're going to see that in these areas here, you will have literally what we call the presynaptic neuron and the postsynaptic. And if you break down the words there, presynaptic neuron, that's just the neuron, all right, the first neuron, that's the neuron before the synapse. And then the postsynaptic neuron is the second neuron. That's after the synapse. Okay, that's basically what that is saying. So the presynaptic and the postsynaptic neurons are bound together, but what we call gap junctions. And these specialized proteins, okay, that help to almost hold two structures together, usually cells, and they actually create a hole there. So we call it a gap. And so it allows the movement of ions, all right, certain substances to move between the cells much easier. Okay, we'll talk about resistance and all that later on. But they're literally these holes that go from one cell to the other, and it facilitates the movement of certain substances, usually ions, to move from one cell to the next. All right. So the nice thing about the electrical synapses is there's no synaptic delay, because we're going to talk about that here when you learn about the chemical synapses. So they're really fast. Think that the synapses are when our signal is going to slow down. Okay. Basically, think of it like this. right? You have a telegraph, if you remember what those were, right? You'd send messages via Morse code, over these electrical wires. And so they would travel really fast over the wire. Where that process slowed down is where the telegraph operator had to listen to the message and start to write it down. Okay, that slowed down that message getting to where it needed to be. Okay, so that's what we'll see in the chemical synapses, because we're going to change over our method of delivery. In the electrical synapses, there's no changeover. Actually, we made it easier. We just stuck a hole. It's like if I'm trying to shout to you from one room to the next through a wall, forget that. I'm going to punch a hole in the wall. Now I'm just going to yell it through the hole. Well, that's what happens here with the electrical synapse. Okay, so it's the chemical synapses that we're going to spend more time on. So let's talk about some of the players in a chemical synapse. Okay, we have our first neuron, which is called the presynaptic neuron. I cannot stress this enough. You need to know these terms. Okay, so presynaptic neuron is going to be the first neuron, and it's going to produce the signal. It's going to make the signal start it up. We'll learn how all that happens later. Okay, so it makes the signal and it transmits it down. And then it's going to transmit that signal to a second neuron. Well, that's what we call the postsynaptic neuron. It's going to receive the signal. And that's what we saw here. Whoops, I'm going the wrong way, folks. That's what we're seeing here. Okay, here's our presynaptic neuron. It's generated the signal, and then it transmits it down, all right, to the synaptic knob here. And then it'll transmit that signal onto the second neuron, which is our postsynaptic neuron. It's going to receive the signal. So we're going to talk about what happens here. All right. So in between our two neurons, we have a gap. And it's a fluid-filled gap. And we call that the synaptic cleft. So what essentially is going to happen is, I'm going to tell you a story here. Now we're going to talk about the events of the synaptic communication. So presynaptic neuron sends its electrical signal down to the synaptic knob. And what will happen is, all right, that synaptic knob has these little bubbles, all right, that are sitting in the synaptic knob. And these bubbles, we'll call those vesicles. And they hold on to what we call a neurotransmitter. Neurotransmitter is a chemical that is going to stimulate or it's going to inhibit, because they can do both, all right, the secondary, the postsynaptic neuron. So actually, the synaptic knob houses these vesicles, these little balloons that have that neurotransmitter in it. And so when the electrical signal comes down and it triggers the release of that neurotransmitter, we're going to see the synaptic vesicles, they'll start to migrate, all right, towards the synaptic cleft on the inside of the synaptic knob. And they'll bind with the plasma membrane there, and they'll release the neurotransmitter into the synaptic cleft there. And those chemicals will diffuse across the cleft, and they will go to the postsynaptic side of the cleft, where you'll have these postsynaptic receptors. And as long as that receptor matches the neurotransmitter, all right, that neurotransmitter is going to bind onto the receptor. And then it's going to initiate what we call a postsynaptic potential, otherwise known as a graded potential. And I'll explain all that to you in a little bit, what a graded potential is. So what we've done is, in that whole process, we've taken an electrical signal and turned it into a chemical signal, and then we'll turn it back to an electrical signal. But where this process slows down slightly is when we go from the electrical signal to the chemical signal, all right, back to the electrical signal. And so we call that a synaptic delay. And you'll see that in chemical synapses, but not so much in the electrical synapses. Okay. I love coffee. Okay, so now that we've kind of talked about presynaptic neurons and postsynaptic neurons, I want to now just focus on the neuron itself, the cell itself, and discuss, all right, some of the components to the cell so we can understand the process of generating an action potential, a nerve signal. So to do that, I need to talk about some of our friends, our transport proteins. Those are our friends. I love proteins. They do so much. And so part of it is we have these special transport proteins called pumps. And the pumps, just like if you have a basement, you have a septic pump, that septic pump is going to pump water out of your basement when it's flooding. Okay, well, the pumps in the membranes here of your neuron, they're going to pump certain molecules, substances, right, in and out of the cell. But here's a couple things that you need to know. One is that these pumps are going to pump things or move substances against, I can't stress this enough, against their concentration gradient. That's a form of active transport. You should remember that from chapter 4. Okay, so these membrane proteins are going to move these substances against their concentration gradient. And by doing so, what will happen is this will cause the cell to maintain a specific concentration gradient. So meaning, for example, there's more sodium outside of your cells than inside the cell. Okay, so these pumps will pump sodium that's inside of your cell outside of your cell. It's going to, but where does sodium want to go? Well, it wants to go down its concentration gradient. So it wants to move into the cell. But these pumps are saying, no, we're not having that. We're going to move sodium against its concentration gradient. Yes, we know that sodium wants to come into the cell, but we're going to pump sodium out of the cell. So guess what? That takes energy. We need energy to do that, to maintain these pumps. So in our membranes of our neurons, you have sodium potassium ion pumps and calcium pumps. And that's what they do. They pump these substances against their concentration gradient. So for the sodium potassium ion pump, it's going to pump sodium out of the cell because there's more sodium out of the cell. And at the same time, it's going to pump potassium into the cell. There's more potassium inside the cell. So it's going to take potassium that's outside the cell and move it into the cell. But understand that there's more potassium inside the cell. So potassium wants to leave the cell. But these pumps don't let that happen. They said, nope, we're pumping you back in. You're trying to escape, we're going to push you back into the cell. And that's what they do. Calcium, same thing. More calcium outside the cell. So these calcium ion pumps, all right, are going to be pumping calcium out of the cell. So thereby, it's going to start to create and maintain a concentration gradient. All right, so those are the pumps. Channels, a little bit different. Okay, so again, channels are our transport proteins. And so here's what happens with channels. They're different now. They're going to allow ions or substances to move down their concentration gradient. Well, good. This is good. So that means that it'll allow stuff to move in and out of the cell, but it'll move down its concentration gradient. Okay, so when these channels are open, because some are always open and some are only open for a brief period of time. When they're open, this is so important that you know this, it's only going to allow a specific type of ion to diffuse. It doesn't let any kind of ion, all right? It lets specific ions. Potassium can only go through a potassium channel. It cannot go through a sodium channel. It cannot go through a magnesium channel or, excuse me, a chloride channel, okay? So when these are open, they can only go through a specific channel that's designed for them, and they always go down their concentration gradient. So if they're going down their concentration gradient, this is going to be a question that you should type your answer into, so get your keyboards ready. If they move down their concentration gradient, do they require energy? Do these channels require energy if they move down their concentration gradient? Yes or no? Oh, I've got somebody already typing in an answer. I love it. Let's see what we got. Please feel free, folks. It's okay if you're wrong. That is correct. Yes, correct. Very good. If it's moving down its concentration gradient, it does not require energy. Very good. Okay. Up the concentration gradient, which we saw here, okay, that does require energy. Okay. So there are three types of channels that I want to talk about today. We've got our leak channels, our chemically gated channels, and our voltage gated channels. All right, so look at the first one there, leak channels. They're always open. These are what I call my 7-Eleven channels. They're always open. All right, so that means that they're going to allow something move down its concentration gradient. So we have sodium leak channels, we have potassium leak channels, and these channels are always open. So remember what I said, there's more sodium outside the cell than inside the cell. So sodium is going to want to go through these leak channels, and it wants to move into the cell because there's less sodium inside. So it wants to go down the concentration gradient. Whereas potassium channels, okay, these channels will allow potassium to move down its concentration gradient. So it's going to allow potassium to go from the inside of the cell to the outside of the cell. I am such a naughty boy. I forgot to start off today's class or this topic with my famous saying. When you think of the neuron, I should say, I want you to think of this phrase, a banana floating in the ocean. Okay? A banana floating in the ocean. So here's our banana. All right, and here are the waves to the ocean. It's floating there. Or you can think of it like the ocean all around it. However you want to decide that, okay? So banana floating in the ocean. What are bananas rich in? Potassium, okay? And then the ocean is made up obviously of water, but what kind of water? Salt water. So when you're thinking of neuron, think of a banana floating in the ocean. The inside of the neuron has a lot of potassium in it. That's where you're going to find the majority of your potassium. Outside of the neuron is where you're going to find your sodium and your chloride. Okay, so salt water. So banana floating in the ocean. That's how I remembered it. Easy enough. Okay, so if you remember that, you'll never forget it. All right, so here's our banana floating in the ocean. Now we have leak channels in it. These channels are always open. They're essentially like pores. And so that means that sodium is going to enter into the cell and potassium is going to leave the cell. So meanwhile, while these leak channels are letting potassium and sodium just move in and out of the cell at their pleasure, we go back here and guess what? Our pumps are continually having to move sodium, potassium against their concentration gradient. So that's like being on a boat in the ocean and there's holes in the boat and you're bailing out the water so the boat doesn't sink. But there's someone in the boat that keeps punching holes into the boat and letting water in. Well, that's kind of what happens here with our leak channels. The leak channels are letting sodium and potassium move in and out of the cell, all right, at their, I don't want to say at their pleasure, but based on their concentration gradient. And then you've got the pumps that are bailing, all right, sodium and potassium in and out of the cell against their concentration gradients. So this is happening all the time. All right, so let's move on to the next type of channel, and that's our chemically gated channels. So when you see this term, chemically gated channels, chemically, this part here, this first word, tells you what we need to open this channel. Because when we're dealing with gated channels, normally these gated channels are closed. And for them to open, we need a stimulus, okay? Something, all right, that is going to trigger the opening of that gate. So the name tells you what we need. Chemically gated channels means we need a chemical. And in this case, we need a neurotransmitter. And so what's going to happen is that neurotransmitter is going to bind on to a receptor there. And then that will cause the gate to open up. and then a specific type of ion can diffuse through the channel. So that means it has to be a specific, all right, if it's a chemically gated sodium channel, then once that gate opens up, the only sodium can go through. Potassium can't go through a chemically gated sodium channel. Okay, it's ion specific, and it tells you. When we go through these, you'll see, all right, you'll be amazed, you'll be blown away. All right, so the other type of channel is our voltage gated channel. Again, it's a gated channel, so that means it's usually closed. We need something to open it up, but in this case, all right, voltage is required, electricity. So in this case, what we're going to see is when the plasma membrane charge changes to a certain value, and we'll talk about this later on, that will trigger this gate to open. And when it does, right, we have a specific type of ion that's going to diffuse through it. So you'll have voltage gated sodium channels, voltage gated potassium channels, all right, all these different types. But again, the title of it tells you how that gate works. Right, so what we're going to look at here is our voltage gated sodium channel. So you look at the name, voltage, okay, that's the stimulus that I need to open up this gate. And when it does open, then this means that only sodium will travel through. All right, so when we're talking about the voltage gated sodium channels, all right, we have two gates. This is a special channel because this type of channel has two gates on it, which means since there's two gates, that this channel itself can exist in three states. And if you look here, you'll see the three states. You'll have it resting, right? You'll have it in the activation state, and then you'll have it in the inactivation state. So those are the three states that we're dealing with. Now, when you look at these pictures here, you'll notice, all right, on the picture, there's lots going on here. All right, we have our molecules on the outside. These are sodium molecules. All right, and then you'll notice we have our transport protein, our channel here with the gates. This type of gate, all right, has two gates. You have the activation gate here, and then you can see it in the background. We have an inactivation gate, which are these two like swellings here. Okay, so there's two types of gates here. And then you'll notice here's our plasma membrane. You can see here's the phospholipid bilayer, but look at this. You have pluses on the outside and negatives on the inside. Okay, so this tells you, all right, this is representing the charge along the plasma membrane here. So normally it'll be positive outside and negative inside. I'll get into more details about that. But for right now, just know the outside of the plasma membrane has a positive charge to it. The inside of the plasma membrane has a negative charge to it. So when the neuron's not doing anything and it's just hanging out and resting, all right, these gates here, these voltage gated sodium channels, are in the resting state, and you have the activation gate closed. So nothing's moving through. So now we get a change in the membrane potential, the charge of the plasma membrane. Okay, so you'll notice here in the next state, the activation state, we've switched the charges. Now it's negative outside and positive inside. But right now, just know that we've changed the charge. And that was enough to stimulate this gate to open up. And so that's what happens. The activation gate swings open. And so now sodium, which we had a lot of it outside and very little of it inside, it now starts to diffuse through. And you know, with diffusion, it will continue until equilibrium is reached. And that's what we see here in this picture. You have three molecules outside, three molecules inside. So diffusion will occur until equilibrium is reached or until the gate closes. So this gate managed to stay open. We managed to reach equilibrium, okay? But what will happen too, in the third state, what we call the inactivation state, the gate will close. But the activation gate stays open, but the second gate, the inactivation gate, closes. And so we are in the inactivation state. So the three states are resting state, activation state, and inactivation state. In the resting state, the activation gate is closed. In the inactivation state, the inactivation gate is closed. In the activation state, all the gates are open. Memorize that, learn that, okay? Because we're going to build upon this, okay? All right, so we talked about the pumps, we've talked about the channels. Let's talk about where we find them in our neuron. So first off, the entire plasma membrane of the neuron, that's basically everywhere. This is where you're going to find our sodium potassium ion pumps, right? These are the guys that are moving things against their concentration gradient. And then we have our sodium leak channels and our potassium leak channels. Now you should know this. You have far more potassium leak channels than sodium leak channels. So therefore, it's much easier for potassium to diffuse across the plasma membrane because we just have that many more. Okay? So keep that in mind for right now. For the length of the entire neuron, you have the sodium potassium ion pumps and the sodium potassium leak channels everywhere. Right. And so this is what's going to help, and we'll talk about what the resting membrane potential is. But for right now, know, right, that the sodium potassium ion pumps help to maintain the resting membrane potential, and these leak channels will contribute to that resting membrane potential. So all this stuff helps with that. And I know that you want to know, what is that resting membrane potential? We'll get there. Okay, so now as we go through the different parts of our of our neuron, we're going to see, all right, on the entire neuron, we'll have our sodium potassium ion pumps and these two leak channels here. They're the whole thing. Now, when we get up here to the cell body, right, with the dendrites, that's called the receptive segment. Makes sense because it's going to be receiving information. Okay, so in the receptive segment, you have these types of channels, chemically gated cation channel. All right, so what's that mean? Well, chemically tells you what type of stimulus you need to open this up. We need some sort of neurotransmitter to open up this gate. And when it does open, it's going to let across a positively charged ion. Then we have chemically gated potassium channels and chemically gated sodium channels. See, these are specific now. Now we're getting real specific. We need some sort of chemical to open up this gate, and once it opens, only potassium can move through, and this one only chloride. So you'll find these gates here in the receptive segment. That's all over the cell body and all over these dendrites here. Then our initial segment is the axon hillock. This is important. This is what I told you, where the action potential, where our electrical signal starts to occur right here in the axon hillock, also known as the initial segment. We are going to see voltage-gated sodium channels and voltage-gated potassium channels, meaning we need a change in the electrical charge across the plasma membrane to trigger the opening of this gate. Once we do, only sodium can move through it. Then we get to the conductive segment. Oh, I'm sorry, guys. Then we get to the conductive segment, and that's the axon, that long, huge process that comes off the cell body. And there we'll see more voltage-gated sodium channels and more voltage-gated potassium channels. Then when we get down to the last part here, what we call the transmissive segment, that's where the synaptic knobs are, we're going to see a voltage-gated calcium channel and then our calcium ion pump. Because remember, I was telling you the calcium pump is constantly pumping calcium out of the cell. And then when that electrical signal travels down the axon and it comes all the way to the synaptic bulb, it'll trigger the opening of these voltage-gated calcium channels, and then calcium can flood back into the cell. So you should really know where all these items are. What is the difference between a chemically gated channel and a voltage-gated channel in terms of how they function? Chemically gated channels open in response to the binding of a neurotransmitter. All right, voltage-gated channels open in response to changes in electrical charge, that membrane potential. All right, this is going to be one of the more important slides for you to know the meaning of what is on here. So this goes back to me saying, banana floating in the ocean. Okay. If you know the banana floating in the ocean, then this slide will make sense to you. Okay. So we have certain substances that are going to be crucial for the functioning of the neuron when we're trying to make an electrical charge. Okay. We're trying to generate that nerve impulse, that nerve signal, the action potential. So we'll see that certain substances will be on the inside of the neuron and certain substances will be on the outside of the neuron. All right, so inside the cell, the cytosol, these three things are going to be found inside the cell. Negatively charged phosphate ions. So a lot of ATP occurs when we break down ATP into ADP. Well, you'll have these phosphate ions just floating out there in the cytosol. Okay, so that's what and they are negatively charged. So we'll see, all right, these negatively charged phosphate ions on the inside. We'll see these negatively charged proteins, and both the negatively charged phosphate ions and the proteins, those are relatively large structures. So they basically remain trapped inside the cell. And then you'll see that we have, all right, a positively charged potassium inside the cell. Okay, so that's going to be on the inside. So that's where we get for our banana potassium. And then outside of the cell in the interstitial fluid, that's where we're going to have our sodium, which is positively charged, and our chloride, which is negatively charged. But together, they make salt. So banana floating in the ocean. Know that. Burn this image into your head. I know it's horribly drawn. I really should hire a graphic artist there to draw a cool picture of a banana. Maybe I'll have my son draw a picture of it. He's really good at art. So that's what we're going to see. We're going to see that more sodium and potassium and chloride is outside the cell, and that and potassium is in higher concentration inside the cell. So that moves us on to the topic of membrane potentials. So we now know the distribution of the substances that are going to help with these membrane potentials. Now let's talk about, all right, how the movement of those substances are going to generate these membrane potentials, these electrical charges. So to start off, I want to talk about what's called the electrical gradient. And the definition of the electrical gradient is the difference in electrical charge between two areas. Now you saw in that one drawing there, way back when, here, right, I made mention how the outside of the cell was positive and the inside of the cell was negative. That's the electrical gradient that I'm talking about, okay? How the interstitial fluid, which is outside, that'll be where the positive charge is going to be found. And then in the cytosol, that's where the negative charge will be found. And so we're seeing a difference in the electrical charges between two areas, inside the cell and outside the cell. Right? So what we'll do is, we're going to add all those up and take the difference, and that will give us that charge across the plasma membrane. And so what we're going to see is normally inside the cell is going to be negative, and outside the cell is going to be positive. And so what the name of that, what we label it as, is membrane potential. And a membrane potential is the difference in charge, all right, between the inside of the cell and the outside of the cell. And that's how we do that, is we just add up all the positive charges outside the cell, all the negative charges inside the cell, and we take the difference. And we refer to that as the membrane potential. It'll have a value, and we'll get into that, okay? And we're going to talk about how we manipulate that membrane potential. Sometimes, if it has most of the time, it'll have a negative charge. So we'll talk about how we make it more negative, and then we'll talk about how we make it less negative or more positive. Okay, so we'll alter, all right, these electrical currents. But for now, keep in mind, okay, inside the cell is going to be negative, outside the cell will be positive. And that difference that we take, okay, is called the electrical gradient. Okay, so now we're going to talk about the chemical gradient or the chemical concentration gradient. Because remember what I said, we have a distribution of substances inside the cell and outside the cell. Those are the chemicals that we're going to be dealing with here. And so when we talk about the chemical concentration gradient, that's going to be the unequal distribution between two areas of these chemicals. For example, right, we saw banana floating in the ocean, more potassium was inside the cell. And we saw that sodium was going to be in higher concentration outside the cell, along with chloride. Okay, so that's our chemical concentration gradient. We'll have an unequal distribution of these chemicals between the two areas. What are the two areas? Inside the cell, outside the cell. And what do we want to see? We're going to see how each substance is going to start to generate its own chemical concentration gradient. And so we know what happens when we have a concentration gradient. Nature likes balance, it likes to be equal. So we'll start to see if these substances can move in and out of the cell, they're going to want to diffuse down their concentration gradient if they can. Right? And when that happens, we'll start to see some changes and some stuff is going to happen and we're going to have fun talking about it. Trust me. So what I want to talk about now is that relationship, okay, with these electrical currents and how that's going to affect these cells. Right? So when we talk about these chemical substances, when we talk about sodium, when we talk about potassium, we talk about chloride, right? They're ions. And we know that ions are charged particles. Okay, if it's a positively charged ion, it's a cation. If it's a negatively charged ion, it's an anion. We talked about that in chapter 2. All right, so we're going to see, all right, how these ions and where they're distributed, how that's going to influence that membrane potential. What's the membrane potential? That's that difference in electrical charge across the plasma membrane. Okay, this is important. I would write this down or star it. A difference in charge distribution at the plasma membrane is termed the membrane potential. Okay, let me repeat that. The difference in charge distribution at the plasma membrane is called membrane potential. And so that has to do with that distribution of those ions. And those ions are charged, and so they carry with them a negative or a positive charge. So that charge distribution, we'll see a difference at the plasma membrane, so we call that the membrane potential. So we talked back in chapter 4 that a lot of these molecules, all right, our plasma membrane is a selectively permeable barrier. It lets certain things in and certain things out. And we said that you can cross right across that plasma membrane if you're really, really small and nonpolar. Well, guess what? That doesn't apply to these ions. So they need help. They need help, and we call that facilitated diffusion. They need facilitated diffusion. And so what's facilitated diffusion? Channels and the pumps. That's facilitated diffusion. Well, not so much the pumps, right? But the channels are going to be the facilitated diffusion. They're going to create these openings through the plasma membrane that's going to allow the movement, all right, of these substances across the plasma membrane. Because without those holes, without those channels, those ions cannot move like they would like to. And basically that plasma membrane is going to create a resistance. It's going to prevent, all right, these ions to moving across that plasma membrane. So that resistance can be manipulated. We can either increase the resistance or decrease the resistance, because keep in mind, all right, the plasma membrane will not let those ions pass on their own. So the only way they can do it is through those channels. So if we want to decrease the resistance, let's open up the ion channels. Hooray! Now we've decreased the resistance. Well, if we want to increase the resistance, let's close them off. Now those ions can't move across the plasma membrane. So Which brings me to a very important point, the last point that I wanted to make on this slide. I'm going to read it to you. Current is generated when the ions diffuse across the membrane through open channels. And when we talk about current, we're talking about an electrical current. So now you have these charged particles, these charged ions. And so when they start to move, okay, they start to generate a current. And so when they diffuse across the plasma membrane, they're going to create this electrical current across the plasma membrane. It's kind of cool. All right. So if you want to decrease that current, all right, then just increase the resistance. Close off those channels and you'll decrease the current. If you want to increase the current, then you decrease the resistance. It's almost like an inverse relationship. Increase one, decrease the other, and vice versa. So there's actually a law, it's called Ohm's law, and basically that's what it says. If you want to increase the current, you decrease the resistance. And in this case, since we're talking about the plasma membrane, okay, if I want to decrease the resistance at the plasma membrane, I open up channels. That'll increase the current. And vice versa. If I want to decrease the current, then I will increase the resistance and I'll close off those channels. I hope you understand that. That's an important concept there. It kind of gives you a little bit of an idea. So when we get these ions moving across the plasma membrane, they start to generate an electrical current. So what is an electrical gradient? Here you go. An electrical gradient is the difference in electrical charge between two areas. If you want to keep it simple, you know, we can say there's a positive charge outside the cell and a negative charge inside the cell. And so therefore that's a difference in electrical charges. So what is a chemical gradient? Great question. Chemical gradient is an unequal distribution of substances between two areas. Banana floating in the ocean. More potassium inside the neuron than outside the neuron. More sodium outside the neuron than inside the neuron. That creates that unequal distribution of a substance between those two areas there. So if any of you are eating a banana today, you'll be thinking about the banana floating in the ocean. All right, just a few more moments and then we'll wrap it up for today, because next week I can still talk about the neurons. So let's talk about what a neuron is doing when it's at rest, when it's just chilling and nothing's going on. Okay, so you should know this, because then we're going to talk about how we excite a neuron, because that's one of its characteristics. It's excitability. And then we'll talk about propagation. Once we've excited it, how we continue to keep that action potential or that nerve impulse going. All right, so in a resting neuron, we are going to see those ions that I just told you about, our banana floating in the ocean. We're going to see how they are unevenly, all right, distributed across the plasma membrane. And again, that has to do with our lovely, lovely transport proteins, but specifically the pumps, those sodium potassium ion pumps, right? And also the calcium pumps down in the synaptic knob, okay? So those pumps are going to make sure that we have a higher concentration of potassium in the cytosol, okay? And it's going to make sure that we have a lower concentration of potassium in the interstitial fluid. And vice versa, it's also going to make sure that we have higher concentrations of sodium outside the cell than inside the cell. But also, all right, the calcium ion pump is going to make sure there's more calcium outside the cell than inside the cell. So that's what these pumps are going to do. So when the neuron is at rest and nothing is going on, no electrical signal is being generated yet, all right, this is what's happening. So like pretend when you're sleeping, okay? So the neuron that goes to your arm muscle, okay? You're not moving your arms, hopefully, while you're sleeping, unless you're rolling over, okay? But that neuron's not going to be doing anything. It's going to be at rest. This is what's happening. So that's the pumps. What about our channels? Our gated channels are closed, okay? They're not going to be open. All right, then we'll see then, because of all of this going on, all right, we'll have more positive ions outside the cell than in it. So that means that gives the outside of the cell that positive charge, and therefore it gives the inside of the cell a negative charge. So we'll see that there is an electrical difference across the plasma membrane. So what gives us, all right, that positive charge outside the cell, and what gives us the negative charge inside the cell? Well, we talked about that with the distribution of the substances there. Okay, for the cytosol, all right, it's going to be relatively negative, and that comes from, remember the phosphate I was telling you about, and also the proteins. They're going to be the ones that really give the inside of the cell its negativity. Okay, so really it's going to be the phosphate containing molecules and the protons inside the cell. All right, so, and then we'll have sodium outside the cell, we'll have calcium outside the cell. That'll help to give some of that positivity, all right, to the outside of the cell. And so now we're going to give a value to our membrane potential. Remember what the membrane potential is? It's a difference in charge between two areas. It's the difference in charge between the outside of the cell and inside the cell. So since we're talking about a resting neuron, we call it the resting membrane potential. You need to know this value. It's been asked on tests before. So the resting membrane potential is negative 70 millivolts, MV. Negative 70 millivolts. Okay. So we actually have found out how to get this value by just using, all right, electrodes. We stick one inside the cell, one outside the cell. We use a voltmeter, and it takes the difference between the positivity outside the cell and the negativity inside the cell. And that's how we get that value, in case people are wondering. I used to have, I don't think I have it on here anymore. No. I used to have a little picture of it. Okay, so what's going on with the, all right, with the cell when it is at rest? Okay, so we now know with the distribution, the uneven distribution of those substances, what makes the inside of the cell negative, and we know what makes the outside of the cell positive. So what's happening at this neuron as it's at rest? Okay, so we're going to start off and talk about what potassium is doing, because potassium is the most, and I'm going to underline this for you. Oh, where is it? It is the most important factor in establishing or setting the resting membrane potential. Okay, so I'll show you why. Okay, so what does potassium want to do? It wants to leave the cell. It wants to leave the inside of the cell, and it wants to go outside the cell. Well, why is that? And you should know this by now. I hope you do. Okay, it wants to diffuse down its concentration gradient. All right, so it wants to move out because there's less potassium outside the cell. So we have leak channels all throughout. Remember what I said, we have many potassium leak channels, and they're 7-Eleven, they're always open. They're always open. So that's what happens. The potassium starts to diffuse out of the cell. It moves down its concentration gradient, okay? And that's nice and all, okay, because it's moving down its concentration gradient. But unfortunately, potassium is limited by the movement, okay? Because as it's moving down its concentration gradient, and that is effective up until a point. But what's going on is, what do we see here? Potassium is positively charged. And what's the charge of the inside of the cell? It's negative, okay? It's negative because those negatively charged phosphate ions are still in there. Those negatively charged proteins are still inside. So even though potassium is flowing out because it's moving down its concentration gradient, and as that concentration gradient starts to equalize, it's going to rush out less. And while that's going on, you're getting those negatively charged items on the inside of the cell saying, whoa, we're negative, you're positive, opposites attract. So we're going to start to pull on you. Okay, we're going to start to slow your diffusion out, and we're going to try to pull you back in. We're going to try to pull it. So think about it. As that potassium leaves, okay, it's going to make the inside of the cell even more negatively charged because those proteins and the inorganic phosphate cannot leave. Okay, so as that positive potassium starts to leave the inside of the cell and it takes its positive charge with it, it makes the inside of the cell more negative. And as it becomes more negative, that attraction, all right, that negative attraction for that positive ion becomes stronger, all right, like the dark side of the force pulling on Luke, you know, to make him do bad things when he's battling Darth Vader, all right? Well, these items will start to pull harder on that potassium, and it makes it tougher for that potassium to leave. So eventually we'll see that ideally, as potassium starts to diffuse out, it's going to try to get to equilibrium, okay? It's going to try its hardest. But as it gets closer to equilibrium, it's going to start to diffuse out slower because the concentration gradient is smaller, but also because those negatively charged items are pulling it back in. All right, so here's the situation. If the only ion that leaked out of a neuron was to be potassium, then our resting membrane potential would actually be negative 90. But that's not the case, okay? So if that were the case, then it would be a different value. But our normal resting membrane potential is negative 70. So now we're going to talk about why it's negative 70, okay? A huge reason for it to be as negative as it is because of potassium diffusing out. Okay, so this is a huge important fact that potassium diffusion is the most important factor, right, in setting our resting membrane potential. If it were the only factor, our resting membrane potential would be negative 90. Okay, but it's not. So we'll talk about why. So when we're talking about why, let's talk about our sodium leak channels. Sodium have far fewer leak channels, but again, they're 7-Eleven, they're open 24 hours, all right, every day. So sodium is going to leak into the cell. So sodium, therefore, is going to influence our resting membrane potential, but not as much as potassium. So here comes sodium. It starts to diffuse down its concentration gradient into the cell. Well, guess what? Remember, the inside of the cell is negative. The outside of the cell is positive, and sodium is a positively charged ion. So it's got two things working in its favor. One is that its concentration gradient favors its movement into the cell because there's less sodium inside the cell. And it's a positively charged ion and has a negative attractiveness on the inside of the cell because of the negatively charged proteins and the negatively charged phosphate ions. So they're actually going to pull the sodium into the cell. So it'll race right into these cells because it has two things working in its favor. And so because of these leak channels, this small amount of sodium being leaked in, all right, we get our value for the resting membrane potential closer to negative 70. If it were only potassium moving in, then it would be closer to this value, negative 90. Okay? Important question. I guarantee you, I shouldn't say I'll guarantee you, but it's a good possibility that you'll see this asked on a test. And you should know why. And then there's one other thing that is going to influence the resting membrane potential, and that's going to be these guys, sodium potassium ion pumps, right? They're not going to do a tremendous amount, but still a little bit. Okay, so we should know that a sodium potassium ion pump is going to move things against their concentration gradient. It's going to require energy, and you should also know the ratio. For every 3 sodium that's pumped out of a cell, 2 potassium are pumped into the cell. Okay, so we're going to push 3 positive charges out of the cell, and we're going to push 2 positive charges into the cell. So 3 positive charges, which are going to be the sodium being pushed out, and 2 positive charges being the potassium gets pushed in. And so that is actually going to, look at that, it's just a small amount to the resting membrane potential, negative 3. And so basically when we add it all up, That's how we get -70. So keep in mind that the resting membrane potential, right, the sodium potassium ion pumps pretty much play a very minor, small role in establishing that -70 value. Right? But, right, what it does do that is very important is that it helps to maintain the concentration gradient for those ions. Okay, that's a very important concept here. It's these pumps, all right, that push the sodium out of the cell, push the potassium into the cell, and maintains that more sodium is outside the cell and more potassium is inside the cell. So by maintaining that concentration gradient, those leak channels and those gated channels will allow the movement of sodium and potassium, all right, down their concentration gradients, which is important. And because of all this, 66% of the energy expenditure of a neuron is used by these pumps. That's a lot, but they're doing it right now. They're doing it 24/7, all day long, every day. That's what they do. Okay. So the resting membrane potential is primarily established by what two structures embedded in the plasma membrane? Sodium ion and potassium ion leak channels. Primarily, though, it's going to be the potassium ion leak channels that play a large role in that. Okay, I think this is a good place to stop. I would love to keep going because I'm really getting into this, but this is a good place to stop here because we're going to really, I feel like if between now and then, obviously after you've studied significantly for your upcoming tests,